Hi, I'm
It
going
takes
to
us
talk
next
to you about the gsDesign Shiny app and the updates we've made to enable uh, easier and more consistent study design practices. First of all, you can see that I'm using gsDesign shinyapps.io, and this particular one is /devel. We also have an R in Pharma version. Here you see the opening screen and inputs on the left. It's a tabbed interface. There are tabs in a lot of places. There's a fair number of commands here, and also tabs on the right for different types of output. Finally, we can do things. One output is tabular. We can do things like change the number of digits that are being displayed. And that gets saved. What I'm going to do here is to restore a design that I've already worked on, which is a really nice added feature. So this is just an R data file that was saved from this Shiny app. So it's basically just restoring the state of the app at the time it was saved. So in particular, there are things that are different here than defaults. Here I've assumed that the alpha for this particular endpoint is 0.0125, one-sided. What we would typically do if it were a single primary endpoint for a trial would be to say 0.025. And 90% power. Here you can see that the trial is doing 5 analyses, 4 interim analyses and a final analysis, and it's setting up various properties, z-values, p-values at the bound, approximate hazard ratio at each bound, the probability of crossing a bound under a hazard ratio, assuming a true hazard ratio of 1, assuming a true hazard ratio of 0.72, which is what we set up the power for. That's basic commands. You can do other types of endpoints: binomial, normal, user specified, and information-based design. This is just an overview, so I won't go through all of them. You can do superiority trials, you can do non-inferiority trials, and we'll also do an example, not today, of a super superiority trial, which is what you would do for, say, a vaccine. The Moderna vaccine used the gsDesign package for its group sequential design. The next thing you do for a time to event is to enter failure rates. There are different ways to do it. This particular design, I'm assuming one-to-one randomization, and I'm assuming a dropout rate of 0.001 per month, and I'm saying an exponential distribution with a median of 12. Now, I could change this and say, well, for the first 4 months, it's going to be something smaller or bigger, depending on what you think. And then for the next 100 months, perhaps go back to 12 months. So you get a smaller median later after people have been treated for a little bit, and perhaps their cancer has had a chance to progress. Next, you can set up timing. Here I've set up timing at 25%, 40%, 70%, and 88%, which ends up being, and in terms of enrollment, I didn't go through that. I'm saying 18-month enrollment duration, minimum follow-up duration of 20 months. Let's make it 24 since we made the time to event a little bit more extended. And so at the end, we end up with 622 patients and 470 events. And that's, about 70% of patients with events, which I think would be typical in a metastatic cancer study. And timing, again, we set it up to be spaced out somewhat in time. So you can see the expected time of these things is 16 months, 19 months, 28 months. So maybe this one's a little bit close. So say you change that to 45%, then it becomes 16, 20 and a half, 27, 35, and 42. You can space those as you wish. Now boundaries, I'll show you a plot for this. These are z-values for boundaries. The top ones are for efficacy, and I've made those in O'Brien-Fleming-like design. We're using asymmetric non-binding designs which basically means you're testing in both directions. But we're testing with an alpha in the bad direction of 0.15 because we'd like to stop early and give up if we see a trend going in the wrong direction, say for mortality, if that's what the endpoint is. And this 1.61, remember the normal distribution, 1.65 would be 0.05, one-sided in the wrong direction. So that's about where we are with these bounds. And typically, the FDA may ask you to use O'Brien-Fleming-like spending for efficacy bounds. So that's how that is set up. And then we get over here to reporting. You've seen plots. You can see different things like the approximate hazard ratio at a time. I've shown you the table. If you want to see a movie and say, well, my design, I've got really fractional events. That's not going to be an issue, but you may not like it. So you could set it up, change your parameters a little bit so that these would round up to close to exact events. So there are different things you can do there. And it's nice to just do this in real time. There, you can also do a report where you can generate our markdown that writes all the parameters for you. And in this case, there's also some text written into the report to describe more completely what's going on. So then if you want to just download the HTML, you can run that. And today's design, and we'll just download that and take a look at it, and you can see the report coming out. And there you can then change the report, like if you want to have GT to write your table out, you could do that here. If you want to delete the plot, here's an extra plot that you haven't seen before, which basically describes the expected pattern for enrollment and event accrual given the assumptions of the trial. Now, also you can go back and you can say, okay, now I want to save the updated design, and I would do that. And that will save on your local machine where you're running the browser. But then you can also just download your R Markdown report so that you can customize it. And you can see in this that when this was run, you also get the run date and time. The R version, we're running 4.1.0, and our gsDesign version 3.2.1. So that's all useful information. I did want to show you one other thing, which is you can now update the design. So if your actual event counts change, let's say you're at the third interim analysis, and I had 100, 150, and 230 events, it'll update bounds for you like that. And then you can also write that report and save it so that you can document how the bound was updated. So I'll stop there, and hopefully that's a useful introduction to the updated application. And we're continuing to work on improving some of these reports and would welcome feedback. So thank you.